Hello everyone. When we consider the usual cell cycle, what you find is there's a period of growth, a phase called G1, followed by a period of DNA replication, where the DNA number, essentially the chromosome number within the cell, doubles, called the S phase, and that's in preparation for a second growth phase of G2 and then cell division known as mitosis. So clearly, for cells to divide, there needs to be this doubling of the amount of DNA that we have in a cell, and that is brought about by what's called DNA replication. Now in this video, I'm going to talk through the evidence for what's called semi-conservative DNA replication. So let's consider what we mean by semi and conservative, the two words at the top here. Well, semi means half and conservative means to conserve, to keep. So essentially what we're saying is when we replicate the DNA, in the molecule that we produce, half of that molecule comes from the parent molecule. Now I've got a big diagram on the screen here that I'll talk through very shortly, and I'll explain what the N15, N14 and all of these bars at the bottom represent. But first of all, let's just consider normal DNA replication. And in fact, this is a very crude sketch of a DNA strand. I have done videos that go into far more detail and depth about this as an actual process, but just as a starting point essentially here, if we imagine a DNA strand, just here we've got a sugar phosphate backbone, and I'll just use the colours red and blue. These red and blue lines represent the bases of DNA, the letters A, T, C and G, and they're base paired by complementary base pairing. So we've got this strand of DNA. What you have essentially is DNA helicase, this enzyme, breaking apart the bonds between these bases. And again, like I said, it's very crude, but we're exposing these free bases on either strand of the DNA. Then within the cell, free DNA nucleotides that have been activated within the cell. So these are essentially like ones that look a bit like this. So you've got nucleotides, which is a bit of sugar phosphate with the attached base. These free DNA nucleotides come and join up with exposed bases via the complementary base pairing rule. So if there's an exposed A, it will join with a T, an adenine, a thymine, a C, a cytosine pairs with a guanine. So essentially we break apart the two strands, free bases then come and join up to the two strands, and essentially what you end up forming are two new molecules of DNA. So essentially all we've done is break apart one strand and copy it. Now the reason why I say semi-conservative, because if you look, this particular strand here, if we take that as the parent strand there, that strand is essentially this one here. And if I use another colour, if I use a sort of, let's say more of a purple one to represent this strand here, this blue strand if you like, that is within this molecule here, because the blue strand, just where I've put an asterisk, that was formed by free DNA nucleotides that came to join onto the red one, if you think of it like that. So essentially what I'm saying here is that in these two DNA molecules, if you look at them both, you can see that half of the strand that makes them up have come from this original parent one. That's what we mean by semi-conservative, that in the strands that you formed, half of the molecule comes from the original parent one. Now what we're going to talk about in more detail is the evidence for this, because essentially, when Watson and Crick worked out the structure of DNA in 1953 using Franklin's X-ray diffraction studies, they favoured the semi-conservative method of DNA replication, but it was one of two possible methods that had to be tested before conclusions could actually be drawn. So what we've had here that I've described is the semi-conservative method, where you've got parental DNA, the parental DNA strands break apart, new nucleotides essentially come and build up on those strands, and we form two molecules. Half of it comes from parental DNA and half of the new strand is essentially new daughter DNA. Now, the alternative idea that was put forward was the simple conservative method. So rather than semi-conservative, the full-on conservative method. And essentially what was believed in that case was that you had this parental DNA, and then somehow you would have two molecules of DNA from that one. One of the molecules was an exact copy of the parental DNA, and another was simply made of daughter DNA, perhaps from all these nucleotides in the cell just randomly aligning. Now, Meselson and Stahl believed that there was no real purpose to this. There was no real reason why these nucleotides within the cell would just spontaneously align. They believed that, and Meselson and Stahl, with two prominent scientists working on this, believed that the semi-conservative method would explain how DNA replicates. It would use the idea of complementary base pairing. So to find out which method was correct, what they came up with was an idea to label the DNA, the original old DNA, if you think of it like that, and find out how it was distributed after replication. So Meselson and Stahl designed an experiment in which they used a few facts to be able to essentially provide evidence for this. Nitrogen has two isotopes, N15 and N14. Now, N15 is known as heavy nitrogen. We'll just make a note here. That's known as the heavy nitrogen. And N14 is known as light nitrogen. So we know that nitrogen has two isotopes, and essentially they realised that bacteria incorporate nitrogen from a growing medium into any new DNA that they make. So when in a medium of a particular type of nitrogen, they will take that up, and they will use that nitrogen to make particular bases. And those bases will then join with essentially a sugar and a phosphate to form nucleotides. So these new strands that we formed would be made up of bases made of a particular type of nitrogen, depending on what the medium was. So this was the experiment, and I'm going to look at the left side of the screen here. So they've labelled the original DNA by growing in nitrogen 15, heavy nitrogen, and that at generation 0 is what we've got just here. So essentially what we've done is we've taken some E. coli, or what they did rather was take E. coli bacterium, grow it in a dish of N15, heavy nitrogen, and then they've extracted the DNA from the bacteria. They suspended it in a special solution and then centrifuged it. And they measured the density. Now you can see clearly here that this line here, this 100%, refers to the density of the DNA being of all the N15 type. So that's how this works. You've got N15 at the bottom of this picture here, N14 at the top. And this graph, if you like, shows the relative percentage or proportion of each of these two types of nitrogen. So we can see that in the first, or zero generation rather, we have 100% N15. Now, if you were to use the picture on the right-hand side of the screen that I drew, I'm going to show essentially using that what happens in subsequent generations. So if we say that this strand here is made of the N15 type, and this strand here has bases made of the 15 type, so we're just going to give numbers to those strands. Essentially what I'm saying are those red dashes that I've drawn, those red bases are of the N15 and the blue bases are N15. So it's very simplistic, but we're just saying that that's a pure N15 strand. Now what they did, Meselson and Stahl, was take some of the E. coli that had been growing and then put that into a dish of N14. And the bacteria would have taken the N14 up. They would have taken that up and used light nitrogen to make the bases for the free nucleotides that it would have in its cell. And then what they did was allow that to grow. And after the first generation here, so we've got generation 1, they would have extracted the DNA, centrifuged, etc. And when they measured the density this time, they found that they got 100%, but of a complete mix. So we've got 100% of a mix. So they knew that there was an equal proportion of N14 and N15 present. So again, let's use the right side of the diagram to show, or essentially to explain what went on. And this is really the evidence for this semi-conservative DNA replication. So if we imagine, again, our DNA strands have split, then this strand again is our 15, and this blue strand over here is 15. Then all of these letters that I have drawn essentially all of these three bases that I drew, the little blues and red nucleotides, they're of the 14 type. Because we said that Meselson and Stahl took some of the E. coli from the N15, or they've been growing on N15, and cultured it in a dish of N14. So if we imagine that those three nucleotides were N14, when our two new strands form, so we're looking at the bottom right of the screen here, this strand would have been of the N15 type, and this strand over here would have been of the N15 type because they're both from our parental DNA. But the strands that we've had attached to those would have been made with N14. So what we can see is in the two molecules of DNA that we've produced, in each of those molecules, half of the strand is of the N15 type and half is of the N14 type. Now, if we had lots and lots of this DNA at the top, it would all replicate and it would all end up like we've got here. It would all replicate, producing DNA molecules, half parental DNA and half new daughter DNA, half N15 and half N14. And that's why Meselson and Stahl observed a complete 100% density after the first generation. So to prove this even more, they went a step further and they allowed it to go for, as we've got at the top here, 14 generations. So they took some of this E. coli that had been growing, then put that into another dish of N14, and they looked at the second generation. Now we can see here that we've got 50% N14 and 50% mix. So just for completeness sake, let's shrink the screen a little bit and let's just explain where that comes from. And that was essentially their key, their evidence for this conservative or semi-conservative method of replication. If we take the two strands here that I've been commenting on, and if we just, again, very simplistically, just take those two molecules, hopefully this is easy enough to understand, and we're saying that this strand is 15 and this is a 14 strand, that's a 15 strand, sorry, and this is a 14 strand in terms of the nitrogen that makes up the basis of it. If we imagine breaking those two strands apart, essentially what we'd have is something that looks a bit like this. We'd have a strand here and a strand here, and then a strand here and a strand here. This strand would be our 15 and this would be our 14, and this strand here again would be our 15 and 14. So all we've done is just break these strands apart. So what we're saying is that between the first and second generation, or after the second generation rather, of growth, to be able to grow and those cells to divide, we've had to replicate the DNA, and we're saying that we've broken the DNA strands here. Now 3 bases, as normal, would come and join up. But remember, this would have been in the dish of N14. Again, not to scale, it's not the best picture, but hopefully you can see where I'm going with this. Again, the diagram's not overly neat, and because that was in a dish of N14, each of these would be of the 14 type, so of N14. So if you have a look what we've got now, in our new batch, so in our second generation, we've now got DNA that is 50% a mix of N14 and 15, and 50% just pure N14. So proportionally, that's what we have here, where I've just drawn the asterisks. And then they continued. So you can imagine if we were to split this one and then this one and this one and then this one, I don't have room to show all those diagrams, but if we split them all, we then get a proportion of a 75% N14 and a 25% mix, and then that gets reduced the more we do that. And all of this essentially provides evidence for semi-conservative DNA replication. So Meselson and Stahl were able to confirm that from an original DNA molecule, when it replicates, half of that molecule is made up of parental DNA and half is made from new daughter DNA. And this was their classic experiment using the isotopes of nitrogen, the N15 and the N14. Okay, hope all that helps.